All right, so our project was on similarities and differences of testosterone and estrogen by Ben Belding, Drew Watson, Jin John, and Thomas McLaughlin. So what is testosterone? It is a sex hormone found in all animals, and more specifically, there are androgens, which are male sex hormones. And androgens are made in the testicles, but more specifically, the Leydig cells within the testicles. It is also produced in the adrenal gland as well. Androgens are primarily made in the ovaries and the adrenal gland, but in much lower doses than males. Normal testosterone levels for men are somewhere around 280 to 1100 nanograms per deciliter, and normal testosterone levels for women are around 15 to 70 nanograms per deciliter. Some things that can cause lower levels of testosterone are age, injury to reproductive parts, cancer treatments, and radiation, or stress. Some things that cause higher levels of testosterone are adrenal and testicular tumors and steroid use as well. Some roles that testosterone plays in the body are sex drive, bone and muscle mass. Low testosterone can also increase body fat. High testosterone can increase red blood cell production. Low testosterone levels can cause fatigue and mood swings. And moving on to estrogen. So estrogen is one of the two main sex hormones that women have. The other is progesterone. In males, estrogen is mainly produced in the testicles, but more specifically the Leydig cells, and also produced in the brain. The hypothalamus in the brain stimulates the cells in the pituitary gland, which causes the Leydig cells to produce estradiol, a type of estrogen. In females, estrogen is mainly produced by the ovaries, corpus luteum, and placenta, but also can be produced by fat tissues in the liver. There are multiple different types of estrogens that are produced. We have estrone, and that's present in the body of females after menopause. And it's a weaker form of estrogen, but can be converted into different types when necessary. Estradiol, both males and females produce this, and is most common in females during reproductive years. Too much can cause acne, low sex drive, osteoporosis, and depression, breast cancer. And too little can cause weight gain and cardiovascular disease. And the third is estriol, which is primarily seen in females during pregnancy. And this helps the uterus grow and prepares the body for delivery of a child. Some things that can affect the production of estrogen are menopause, extreme dieting, obesity, age, pregnancy, and strenuous exercise. And some roles that estrogen play in the body are to help the growth of the egg follicles, which help to increase fertility for females, and the thickness of the vaginal wall, and maintaining the mucous membrane that lines the uterus, formation of breast tissue, maturation of sperm in males, and sex drive for males and females. So what are testosterone and estrogen made of? They're both made up of a cholesterol backbone, and cholesterol is a steroid, so steroids are lipids. And due to them being hydrophobic, they do not resemble lipids because it was composed of like 4 fused rings, as you can see here. And cholesterol is the most common steroid and is a precursor to making vitamin D, cortisol, bile salts, and of course, testosterone and estrogen. Cholesterol is a waxy substance that helps the body make cell membranes, vitamin D, and sex hormones. It comes from 2 sources: the food you eat and your liver. Cholesterol, as well as other fats, are carried in your bloodstream as spherical particles called lipoproteins. And the most common lipoproteins are low density LDL and high density HDL lipoproteins. LDL cholesterol can contribute to the formation of plaque buildup in the arteries, increased risk of stroke or heart attack, and you typically want your lipoproteins to be somewhere around 70 milligrams per deciliter. HDL cholesterol is the cholesterol you want for your body to be in good health. You can achieve this by exercising like every week and not smoking, losing weight, and just doing healthy things, and also avoiding saturated fats. And you want your levels to be around 50 milligrams per deciliter. And the cholesterol, it's circulated into the Leydig cells, ovaries, adrenal cortex, and liver. It begins the synthesis of testosterone and estrogen. And these are all important enzymes in the production of testosterone and estrogen. And the first one is CYP11A1. And this is a gene that encodes cytochrome P450's superfamily of enzymes. In cytochrome P450, proteins are monooxygenases, which catalyze reactions in the drug metabolism and the synthesis of cholesterol. The protein is located in the mitochondrial inner membrane and catalyzes the conversion of cholesterol to pregnenolone, which is the first rate limiting step in the synthesis of steroid hormones. CYP17 is another enzyme, and this is also a gene that encodes the cytochrome P450 superfamily. It is a microsomal enzyme that catalyzes two different activities, 17 alpha hydroxylase and 1720 lyase. It meditates 17 alpha hydroxylation of pregnenolone or progesterone to yield 17 alpha OH pregnenolone or 17 alpha OH progesterone. It also mediates cleavage of the C17-20 bond of these compounds to yield dehydroepiandrosterone, which is also called DHEA, or androstenedione. You also have CYP19, which is a gene that encodes for cytochrome P450 superfamily enzymes, and codes to make an enzyme called aromatase, which is used to convert androgens such as testosterone and androstenedione into various forms of estrogen. You have 3B-HSD, and it works in the biosynthesis of progesterone from pregnenolone, where you have the 17-alpha hydroxyprogesterone from 17-alpha hydroxypregnenolone, and androstenedione from dehydroepiandrosterone, DHEA, in the adrenal gland. This also is the only enzyme that is not a part of the cytochrome P450 family. And finally, you have the 17B-HSD. That's an enzyme that catalyzes the final step in the bioactive anabolic steroid production by producing testosterone. So how testosterone is made, you have cholesterol to pregnenolone. So within the mitochondrial, cholesterol is converted to pregnenolone by the CYP11A1, which is located in the inner membrane. And you have 17-alpha hydroxypregnenolone to the DHEA, and the enzyme CYP17 catalyzes the 17-alpha hydroxylation in the 1720 lyase conversion of 21 carbon steroids to 19 carbon precursors of sex steroid. And you have the DHEA to androstenediol, and it can be converted that by the use of the enzyme 3-beta-HSD, which catalyzes the reaction within the adrenal gland. And then finally, androstenediol to testosterone, which is converted through the use of that 17B-HSD enzyme, which catalyzes the reaction to get to testosterone. And how estrogen is made here. Oh wait, I'll go back. This is just a chart that kind of shows all that. So how different forms of estrogen are made. So you have testosterone, estradiol, and to produce estradiol, you have to go through the whole process to get testosterone. So the CYP19 gene comes into play and codes for aromatase, which then catalyzes testosterone to produce estradiol. Then for estrone, you have androstenedione to estrone. So it's made from that androstenedione, the CYP19 gene codes for aromatase, which then catalyzes the reaction for androstenedione being made into estrone. So finally, estrone to estradiol to estriol, and it's made from either estrogen or estradiol from the use of the HSD17, which catalyzes the reaction. And here's another graphic that's kind of complicated and shows all the different pathways here. So here we have the 3D structure of the estradiol receptor. The hinge region, which is shown in blue here, is the D domain, also known as the hinge region, and it has a nuclear localization signals, which is seen here where the ligand is binding. It is a flexible region which connects DBD and LBD. And you have the F region shown in gray. The F region modulates gene transcription in a ligand-specific manner. the ligand binding domain, shown in yellow, and that contains a hormone binding pocket and is responsible for most part of the functions activated by ligand binding, such as regulating the binding to AF2 and activation function. And the DNA binding domain, shown in green. The DNA binding domain binds to the estrogen response element, which plays a huge role in dictating the binding affinity. And here's the 3D structure of the androgen receptor. So you have the different components, the N-terminal domain, DNA binding domain, ligand binding domain, and hinge region. So kind of similar to the last slide. So it's just some structural differences in how they affect function. So the main hormones are classified as estrogens, and estradiol and estrone are two estrogens that are structurally very similar to testosterone. So one difference between the two is that estradiol and estrogen contain one less methyl group than testosterone on carbon 19. And the other difference is located in the A ring, which is fully delocalized in both estradiol and estrone, meaning that the electrons are distributed equally in the ring, opposed to the A ring in testosterone, which is not delocalized. The structural differences between testosterone and estrogen, such as estradiol and estrone, allows the steroids to bind to different receptors in the body. And the testosterone binds to the intracellular androgen receptor in target tissues, which are located in many organs, including the testes, ovaries, bone, muscle, skin, liver, and myeloid tissues. And estradiol and estrone bind to estrogen receptors known as ERs. Estrone prefers to bind with the alpha estrogen receptor. So we looked at KD binding affinity as our quantitative descriptor. And KD, which is the equilibrium dissociation constant, can be calculated by measuring the rate of dissociation between androgenic hormones and androgen receptors. An experiment we looked at was conducted by Elizabeth M. Wilson and Frank S. French. They focused on observing the KD values for testosterone and the androgen receptors in 3 different tissues in rats. And these tissues were the tissues from the testes, prostate, and epididymis. And the results showed that the KD value was lowest for testosterone centralized in the epididymis at a 2.5 times 10 to the negative 10, and highest in the prostate at 4.9 times 10 to the negative 10. And the testosterone produced in the testes was slightly, but not much higher than testosterone produced in the epididymis, with a value of 2.9 times 10 to the negative 10th. And this data indicated the binding strength of testosterone to the androgen receptor is the strongest in tissues located in the epididymis and weakest in tissues located in the prostate. In the weak binding affinity in prostate tissues between testosterone and androgen receptors, and one hypothesis as to why prostate cancer becomes more likely as males age and have more unbound testosterone molecules in the prostate. And here's the works cited pages we have. We have a lot of sources. And that's it for our project.